Okay, welcome back to this review of enzymes, and this hopefully will help you prepare for any exams that you may have coming up and have a better understanding of what enzymes are. Okay, first thing is that you have to know that proteins are organic, um, and we look at the elements that are in all proteins, and that's carbon, hydrogen, oxygen, and nitrogen. And the thing you got to remember is that it's the way I remember it is you can spell the word John. If you remember your other elements. Um, so remember that for proteins. The other thing is they're organic catalysts. And what does that mean? A catalyst is anything that speeds up a chemical reaction. And they're organic, meaning that they have carbon and hydrogen in them. So the Regents loves to refer to them as organic catalysts. Sometimes they talk to them as biological catalysts. Either way, they're talking about an enzyme. Okay. All right, so let's take a look at what they look like. Well, if you remember, this purple molecule right here has this interesting shape, and we know that proteins have a very specific shape, and enzymes have this shape right here. And they all look different, but the specific enzyme, this shape is called an active site. And what the active site allows to happen is that you have a substrate which actually fits into this active site. We talk about this induced model where it actually changes the shape around it a little bit, but it does fit into this enzyme. So we get what's called this enzyme substrate complex. And so we have this substrate with the enzyme, and so as they bond together, it's the enzyme substrate complex. So the chemical reaction occurs. You can see that they're breaking up these two monosaccharides, basically. We also need to add water to this. So we know that as this adding of H2O is hydrolysis. And so this is what's considered to be a hydrolytic enzymatic reaction. And we can see that our two monomers now are separated from each other. So that might be glucose and fructose. And you can see that they're released from the enzyme. So that's an important concept too that you have to realize is that once the substrates are released or the products are released, the enzyme is available to be used again. So they can use again and again and again. Nice thing is you don't have to make new enzymes. Okay, so this enzyme can come back, another sugar, another disaccharide can bond to it, and again, this process can repeat itself. Okay, so the next thing is we're going to talk about is what can affect the reaction rate of an enzyme. And we've been looking at temperature, pH, and surface area. There are some others like cofactors and inhibiting factors, but right now we're going to focus on these three factors. And we've looked at those under lab. And first thing we'll look at is enzyme and pH. So one thing you should always remember is this pH scale. So when you're dealing with a pH of less than 7, we're talking about acids. When we're talking about a pH of greater than 7, we're talking about bases. So all the way up to 14 being the strongest base, all the way down to 0 being the strongest acid. Now if we look at this chart, this optimum pH, the pH that this enzyme works the best at, we can see that pH of 8 is its optimum pH. And you can see that if we go too basic or too acidic, even back to neutral even, you're going to see that this enzyme loses its effectiveness. That's what it's showing. Okay, here's another graph of pH, and this is showing you two different enzymes in two different locations. And how can you tell that? Well, this enzyme has a optimum pH of 3. And this enzyme has an optimum pH of 8. So where would they be found in our body? Look at this chart, it becomes obvious. You can see that the blue it would be found in the stomach with a pH of about close to a 3. And you can see that this one with a optimum pH of about 8 to 9, it's going to be found somewhere in the intestines. That's the way you really need to kind of determine where they are in the body. Remember, the stomach is acidic, so their enzymes need to be able to handle those acids. Okay, so we can look at here's just another picture of an enzyme, the way the Regents kind of draws it sometimes, and you can see that it has a specific shaped active site. And you can see that the substrate is going to match that site. Okay, so this could bond in there and then break apart, and you can see that you have two products. Maybe you have a glucose and a fructose or something like that. Okay, all right. Moving on to temperature. Temperature, you can see, has this optimum temperature. And say if it was human body, we could see that that would probably be 37 degrees Celsius. And if we go higher than that, you can see a real steep drop off in the reactive rate. Why is that? Well, high temperatures tend to denature the enzyme. It means it loses its shape, and then it loses its effectiveness. If it goes the other way, if we see this lower temperature, it doesn't necessarily denature the enzyme. What it does is it slows the molecules down so that there's less substrate making contact with the enzyme. So you have less enzyme substrate complexes, and then the reaction is slower. The last thing we're going to look at is enzymes and concentration of substrate. If we look at this, we can see that as we're adding more substrate, our reaction rate's going up and up and up and up and up, all the way to here. This is the point of equilibrium, or really it's really the point of saturation. Basically, it saturates all the enzymes. All your substrate saturates all the enzymes. And how is that possible? Well, if you have a certain amount of enzymes, you have a certain set number of enzymes, well, we're going to add our substrates, and it can handle more substrates. But then as you keep adding more substrates, more and more of your enzymes are taken up by the substrate, basically binding to the substrate. And get basically this level of saturation where all your enzymes are bound up by substrate, so that you can't bind any more substrate to any more enzymes. There's none that are available. Or basically what happens is that you have to have one of these released before another sugar or another disaccharide could be broken down. That's more realistic there. Okay, well, this concludes our enzyme video. I hope you've learned something, and I hope this makes enzymes a little bit more clear. Okay, and thank you.